Next up is Anna Levina. Anna graduated from Duke University in 2007 with a bachelor's degree in biology, concentrating in marine biology, and followed that with a year-long internship at, am I saying this right, Aris Tuberculosis Foundation? Vaccine Foundation. In 2010, she received her master's degree in oncology from the University at Buffalo, Roswell Park Cancer Institute Division. She then joined Cornell University Department of Plant Breeding and Genetics to work on her PhD. She's currently hoping to finish her degree at the end of the year. So let's welcome Anna Levina.
Okay. Hello, everyone. Thank you very much for inviting me to come here and be able to talk to you about my project and dealing with network analysis, using network analysis in potatoes to try to take a very complex data set, make it more manageable, and apply it to real-world phenotypic characteristics. But first, because I don't get to talk about potatoes that much, I want to tell you why we actually should study potatoes and put more money and funding into it. The main thing is people eat a lot of potatoes. Average consumer, according to the Idaho Potato Board, eats about 112 pounds of potatoes. That is compared to about 180 pounds of vegetables that they eat per year. So potato is one of the more, if you count it as a vegetable, as one of the more important vegetables people consume. And they usually consume it in both, so for frozen, that's usually your French fries, whether you eat at home or McDonald's. People consume 35 pounds of fresh potatoes. That's in your mashed, boiled, baked. They also eat about 15 pounds of chips, which is what, for our breeding program in New York, we focus mostly on chips and fresh market, partially because that's where the chip industry is. Also, potato chips were invented in New York, according to the urban legend, and it's a very important part of our breeding program. Now, in addition to being able to get starch and calories from potatoes, people are actually able to get other nutrients. For example, for a medium-sized potato, you can get 45% of your daily vitamin C. If you actually look at a grocery bag of potatoes, they will often tell you it's a high potassium food. You can get as much potassium from it as bananas. So in this country, actually, the sad part is, people get their vitamin C not from citrus, but oftentimes from potato snacks. But that is why it's important to study what is actually what metabolites, what nutrients are in potatoes, and how can we improve it, because people just eat a lot of it. So for our breeding program, as I mentioned, we focus on chip quality. We also do look at for tuber appearance. So for our fresh market potatoes, when you go to the grocery store, you usually select the prettiest potatoes, like a beauty pageant. You look for the smoothest, whitest, blemish-free potato, at least in the U.S. Europe doesn't care, we peel our potatoes. And we also look for disease resistance. But for chip color, what we select for is starch composition, starch levels, and fry color. So what that means is we make potato chips. If you ever come to Ithaca in about between January and February, we fry potato chips every single day. And we want potato chips that look like this. Very white, kind of, I mean, it's a really washed out picture, but we want as white potato chip as possible. What we don't want is potato chips that are black. One, they taste bitter. Some people do like that, but on average, people don't want to have potato chips that look like this. Also, the industry is really worried about acrylamide, which is a carcinogen. So, and the darker the potato chips, the higher levels of acrylamide you usually have. So we try to breed potatoes that are potato chips that are as white as possible. But it's not as easy. So there's over 1,000 metabolites of potato. Potato is also an autotetraploid and is very heterozygous. To give you an example, there's a SNP about every 15 to 20 nucleotides. I've had a population where I've crossed 2 resistant parents for a disease called common scab, which is you can see in the corner, and we had a very nice bell curve shape of spread of both resistant and susceptible varieties. And the biggest thing is most breeding of potatoes done phenotypically. As I mentioned, we walk the fields. So the digger pulls up all the potatoes to the top, and we walk the fields, and the potatoes have about 15 to 20 seconds to make an impression. It's the ultimate of speed dating. And for disease resistance, we also walk the field, and we look at, we try to pick out potatoes that don't have the scab, which is a big problem in New York. But oftentimes you have problems such as population not germinating. Deer will eat your potatoes. So it'd be really nice if we could make, breed, and select, not just phenotypically. So one of the solutions is you have this very complex data set, these thousands upon thousands of metabolites. You can use network analysis, and I'm going to be using WGCNA, which is weighted gene correlation network. And I'll talk more about further in my presentation. But it's been used in it's mostly for gene expression, but it's been used for metabolites in both corn and tomato. And what it does is it takes all this lots of these complex metabolites and organizes them into groups. And then you can use these groups to connect them to real-world phenotypes. In our case, it would be chip color. And then we can select for both chip color and maybe yield and maybe appearance and be able to juggle these objectives more simultaneously. Interesting. That did not go like that on my screen, but okay.
Sorry about that.
I actually looked like fine this morning, too. You screwed it up. So, for experiment design, I had a diversity panel of 200 diverse potato varieties. We used the SolCap panel. We grew them over 2 reps, and we took 3 tubers for each line, for each clone. We took 2 cores per tuber. We used a cork bore, as you can see here. We then cooked the cores in the autoclave, and the reason we cooked them is because, for the most part, people eat potatoes cooked. A lot of metabolist studies will look at raw material, but people don't eat raw potatoes. So we had to make sure we cook them. And we extract them with a methanol buffer, and then we'll quantify them with a UPLC mass spectrometer. All right. So as I mentioned, I used a network analysis. So we had this very large data set. We had about 981 raw features that we found. And we used WGCNA. And what it does, this is an example of a WGCNA network from tomato. And so what it does is you have these groups, and the WGCNA groups them into modules, and it calls them by color. So you will see me refer to them as the blue module or the red module, the black module. I mean, you could call them by presidents of the U.S., you can call them animals, it doesn't matter. It's an arbitrary name that has nothing to do with what's in it. But you can see that, for example, in the blue module, you can have fructose and glucose that is really correlated with each other and correlated with other compounds. And you can see that the light blue module is connected to the red. And again, this is in tomato, this is not my data. Just kind of give you an example of what is the goal of this. We want to, instead of taking these 981 features, we want to cluster them into groups. And so we've done that, and just kind of the data flow is I started with 981 features. The data was not normal, so we natural log transformed it. We've then calculated BLUPs, partially to account for biological reps, so we had 2 reps, and we also had 2 technical reps. And then those BLUP numbers were used for both WGCNA and GWAS. So I've mentioned linking this to real-world phenotype, and we'll go more into depth on that. But we had 27 modules. Again, they're named by a random color. The color has nothing to do with what's actually in it. And so then we looked into real-world phenotypes such as chip color, glucose and sucrose concentration. And there were 27 modules, and things that are not correlate with each other or with anything, they end up in a gray module. I only had 6 features that end up in there, so I can actually make predictions and explanations about more than 90% of my data. To kind of zoom in on this, so we have a heat map of modules and real-world phenotype data. It's just chip color score. So if you remember, low chip color score is better. It's a whiter potato chip. And so you can see that you have the blue module, which is a very strong correlation to what I call the bad chip color score. And then you have the black module, which is what I will be talking about more, which is correlated with the good potato chip score. So we wanted to also look at genetic explanation. So we did GWAS, and we did it at both the module level and individual features. So for the module level, for the phenotypic data, I've used the module eigenvalue. For the individual features, it was again the BLUP score. For our genotype data, we did the SolCap Infinite chip, which has 3,800 markers, which seems like a very low number, but the thing to keep in mind is that LD is huge in potato. We breed an F1. They were saying that the difference between the first cultivated potato and what we have now is about 8 to 10 meioses. So there's very heterozygous, large AD, 3,800 marker will cover the genome. So when I did GWAS, so remember this is the black module, I know that the color is green, but this is talking about the black module that is correlated with the good chip color. And the red line is the bond priority correction, which is much stricter than the FDR. And you have strong hits. You have markers that are hitting in chromosome 3 that explain a module. And when I looked at this on an individual level, so this is for feature number 502. This is an example for a Manhattan plot for feature 502, which is in the black module. You have markers. They're on the same chromosome, similar region. And when you looked at this, there were 19 out of the 31 members of the black modules that were significant at above the bond priority correction, and they were all on the same locus. And so in conclusion, I was able to use WGCNA to take this complex set of 981 features and cluster them in 27 modules and make correlations and hopefully predictions with important phenotypic traits. We also found strong genetic explanations on both module and individual features. So my future goals is to be able to identify what's in them. This was an untargeted profile. We do not know what any of these things are. That is why I'm working with hopefully some chemists at Oregon State and maybe even some chemists at Texas A&M to be able to identify what these things are. So I would like to thank my PhD committee, my advisor Walter DeYoung, who has been extremely helpful in this whole endeavor. My committee, Dr. Michael Mazurek and Dr. Keith Perry. Dr. Owen Hakanga is the person who originally pioneered this method in corn and has been extremely helpful with this whole assistance. His tech, Dick Kazmar, helped me with extractions of these cores and going through the whole cooking and the methanol extraction process. Corey Bruckman at Colorado State processed the initial raw data. And hopefully Dr. Patrick Vido and Donovan at Pressa would be the chemist collaborator at Oregon State that will help me identify these compounds. And it's my lab. And are there any questions? Yeah.
So do you think the length of cooking might have affected your result?
It's very possible. I mean, so the way we decided how to cook is we cooked the cores up until the point when you pierce them with whatever sharp instrument you had, like a knife, it will go through. Sort of like you figure out how to cook a baked potato. So we had a lot of trial and error of how long do you cook them in autoclave so they get to that point. My entire lab and floor smelled like baked potatoes for a while. But it's definitely like you definitely, if you want to replicate this, you want to do it in both cooking, like steaming, frying, boiling. We didn't have a way to boil this many samples all at once. Yeah.
So you have those 981, I guess, what the features?
I call them features because it includes things that were very similarly, like they had the same M over Z value and retention time, and so they got clustered together.
So what do you hope those, so you have some that are highly correlated with a positive potato chip. What do you hope that those are? What do you think that those
I think
features
that
are?
some of those are probably potential glucose and fructose. We also, for good chip color, it is very possible that we have some compounds that are glycoalkaloids, the bitter compounds. Because one of the parents of a lot of the modern chipping varieties is Lenapede, which was released and chips beautifully, and it's extremely bitter. So it's been banned to be like read for, talked about, like they removed the name. But we don't select when you chip, you just select on the things that make it a white potato chip. We don't select if it's bitter, so it's possible we actually dragged up a lot of those glycoalkaloid compounds. Yeah.
So when you do our GWAS analysis based on the modules,
yep.
Each individual is going to have a Eugen value for...
Yeah, so each... Yeah.
Okay, gotcha.
So couldn't you use like principal component analysis and biplots to group them?
I mean, you could, but it doesn't group them in like as many modules. Like when you actually, so you could. You can do PCA. There's no population structure, but this allows you to group on further levels than just PCA.
So you used one compound to extract these metabolites. I don't remember what.
Methanol.
Methanol. So what do you think you'd find if you used water or benzene or some other?
Well, you'd be able to pull, like, if you would use actually an ammonia treatment, we'd be able to pull some of the carotenoids. So we use methanol because we could pull the polar and some of the more polar compounds. But you definitely, if you use a different extraction, you'd be able to pull things that are able to dissolve in that.
Do you think you'll do that at some point, or some?
Someone. I'm really hoping that I'm not the one doing it. I just want to know what these things are. They can write the papers and graduate. Yeah.
So on your Manhattan plots, and you have your grouping. Your grouping is full.
This one?
Yeah, either one.
Okay.
So you say that has like 33 compounds possibly in there?
It has 31, yeah, it has 31 members.
So if those are biochemicals, you would expect there to be several pathways that affect it.
Yeah, it's possible that it's several pathways or there might be all controlled by the same transcription factor.
Okay, but this is related to my research too, because most likely these are quantitative traits that have multiple genes expressing them. So why do you think you're only seeing one peak?
For this one, there is one of the invertases is on that chromosome,
Okay.
and there was a GWAS paper that looked at just chip-in varieties, and they found a strong hit also on that chromosome in that similar region. So we're thinking we picked up something that is controlled by that invertase enzyme.
So, but if you look at it, you don't have anything else anywhere close, like no other peaks.
I mean, you have, yeah.
Do you think this is I have the same results in my research too, that we have one peak,
Right.
and there's no other peaks close. So it's just because that peak is so significant that it's washing out the rest, and if you put that into your model, you'll find other peaks. Or do you think that this?
I mean, I think it is so significant because it's basically what directly, is responsible for the path for these metabolites.
So do you see this similar trend of one peak in your other groupings?
Not everywhere. I mean, so in terms of peak, we've seen significant peaks at chromosome 3 and 7, but when you look at significant peaks, they're kind of other ones. They are spread all over.
Okay, so you have other groupings that have like more than one significant.
Yeah, I do.
Great presentation, Anna. Thank you very much.
Thank you.